Hi student, in this video lecture we will talk about one of the important topic, that is about the ribozyme. As we know that in our body most of the reactions are biological reaction occur in our body. Now these reaction it is proceed are catalyzed by a fructineous molecule. That molecule we called it as enzyme. Now enzyme are biologically made from the fructine. Now we have an enzyme that is not made from the fructine. So that enzyme that is called is ribozyme. It is one of the exception to the fructineous nature of the enzyme, that is ribozyme, because it will made from the RNA. So that's why it is the exception to the proteinous nature of the enzyme. So let's get started from the ribozyme. Now the first point, that is about the etymology of the ribozyme. If we look to the term that is riboenzyme, so we will get 2 points, two term. The first one that is ribo, so which mean that the ribonucleic acid mean RNA, while the second term that is zyme, which is actually taken from this enzyme. Actually this RNA it will act as an enzyme, so that's why the term that is given to ribozyme. The ribo term it is taken from the RNA side, while the zyme term that is this is taken from the enzyme. So the term that will finally we get, that is ribozyme. Now we will talk about the discovery of the ribozyme. So the ribozyme was discovered by two different scientists in a different experiment, Thomas Sich and Sidney Altman. He performed experiments separately and concluded that the RNA have a catalytic property. So these two scientists separately discovered the ribozyme, and he concluded that the RNA have a catalytic property. Now the term that we use familiar with it's the term ribozyme. Now who proposed the term ribozyme? So this term is actually proposed by or coined by the Kelly Kruger. This is a scientist which gave the name that is ribozyme. Actually the discoverer said it this have an catalytic property, but the name ribozyme this was proposed by the Kelly Kruger scientist. Now we will define the what the ribozyme is. So you can see here that this is the definition of the ribozyme. Actually RNA enzyme that catalyze a chemical reaction. As we know that the enzyme that is proteinous in nature, that we have already discussed in the introduction. So enzymes are proteinous in nature, and these enzymes are used to catalyze a specific chemical reaction. Now in case of the ribozyme, this is actually RNA. When RNA is used to catalyze a chemical reaction, now this enzyme this RNA is now called as ribozyme. So this is the first definition of the ribozyme. Now let we discuss the second definition of the ribozyme. RNA molecule that is capable of performing a specific biochemical reaction similar to the action of the protein enzyme. Mean this RNA is actually act as an enzyme, and this RNA is capable to perform or to performing a specific biological reaction. Mean it is used to catalyze a biological reaction as the same way as enzyme a proteinous enzyme act. So in the same way this RNA molecule will catalyze a biochemical reaction. So this is the definition of the ribozyme. Now where this ribozyme is present? Now if we talk about an cell, so it is present in nucleus as well as in the cytoplasm, and the cytoplasm it is related to the organelles. So this is about the occurrence of the ribozyme. Now we will talk about the smallest ribozyme and largest ribozyme. As we know that the ribozyme is actually the RNA, so it must be made from the nucleotides. So on the basis of the number of nucleotides, the number of nucleotide is actually vary in the ribozyme, but only on the basis of this number of nucleotide we will decide. So this is the smallest ribozyme and this is the largest ribozyme. So this decision we will make on the number of the nucleotides. Now you can see here this is a sequence GUGGC. It is located to the 3' end of the RNA. So this sequence it is actually act as a ribozyme, and this sequence it contains 24 nucleotides, and this sequence is actually act as a ribozyme, and this is the smallest ribozyme, as it contains 24 nucleotides. So this is about the smallest ribozyme. Now, why is satellite ribozyme mean? We as ribozyme, it is actually the largest ribozyme because it contains 154 nucleotides, large number of nucleotides that is present in the VS ribosome. So it is the largest ribozyme. Now we will discuss this what property are responsible for the RNA to act as an enzyme. So this RNA is act as an enzyme because it have two important property. The first one that is it have a long length. The next property that it have three-dimensional structure. So these two property it is responsible for RNA to act as an enzyme. So why this RNA act as an enzyme? Because of these two property, and these two property will enable RNA to act as an enzyme. Now what is the difference between the ribozyme and a typical enzyme? So here we will discuss the difference between these two, ribozyme and enzyme. So the first point that you can see here, it is an RNA composition. All the ribozyme it will match from the RNA, while in case of the enzyme, all the enzyme that is made from the protein. So the nature of these two are specifically different, as this is made from the RNA while the enzyme it is made from the protein, a biomolecule that is protein. Now you can see here it is mostly present or related to the organelle that is ribosome, while the enzyme that is all present throughout the cell, including various organelles as well as in the nucleus. So you can see here the two difference that is mainly present between the ribozyme and enzyme. The first one that is the composition, and the second one the occurrence. So these two are the difference that is present between the ribozyme and enzyme. Now what is the similarity between the ribozyme and enzyme? Now you can see here that both of these, as the ribozyme as well as the enzyme, both of these are used to catalyze a specific biochemical reaction. Both are used to catalyze a specific biochemical reaction. Now the second point that is both are not used in a reaction, as we know that it catalyzes a biochemical reaction but does not take part or use in a reaction. It is remain unused at the end of the reaction. So this is the two similarity that is present between the ribozyme and enzyme. Now here we will discuss the characteristics of ribozyme or property of ribozyme. The first characteristics that is enzyme that use RNA as a substrate. As we know that it is actually an RNA but act as an enzyme, so that's why it is an actually RNA enzyme that use RNA as a substrate. So that's why it is the first characteristics of the ribozyme. The second characteristics that is an enzyme that synthesizes RNA primer during DNA replication. When another DNA it is formed from the parent DNA, this process is called as DNA replication. Now this replication process it is initiated by a primer, RNA primer. Now this enzyme, this ribozyme, it is used to synthesize this main product for the DNA replication, and that is RNA primer. So this is the second characteristics of the ribozyme. Now the third one that is an enzyme that catalyzes the association between large and small ribosomal subunits. As you know that ribosome it is made from the two unit. One is called a small unit while the other it is called as ribosomal small unit, and the other is large unit. Now to catalyze the association between the small and large unit of the ribosomal, this is done by an enzyme, and this enzyme that we call it is an ribozyme. Now the last characteristics that is a ribozyme is an RNA molecule with a well-defined tertiary structure. As we know that this ribozyme is actually the RNA molecule, and that have a 3D structure at tertiary, and it occupy the tertiary structure. So here are some of the characteristics of the ribozyme. Now, on the basis of their occurrence, or it is present naturally or it is synthesized in the laboratory. So on this basis we will divide the ribosome into two categories. The first one naturally occurring ribozyme, and the second one that is artificially synthesized ribozyme. So those type of the ribosome that is found naturally, so such type of ribosome is called as naturally occurring ribozyme. Here are the example of the naturally occurring ribozyme. The first one that is hairpin ribozyme, peptidyl transferase, it is an also an ribozyme and naturally occurring ribozyme, while the third one, that is hammerhead ribozyme, it is a naturally occurring ribozyme, while those ribozyme which is synthesized in the laboratory, this is called as artificially synthesized ribozyme. That we have the two example, that is allosteric ribozyme and ligase ribozyme. This is the example of the artificially synthesized ribozyme. Now, what is the function of ribozyme? So we have two functions. The first one, that is catalysis of biochemical reaction. As we know that ribozyme is actually the RNA. Now this RNA is used to carry or to catalyze different type of biochemical reaction. As we have certain biological process, this is maturation of the transfer RNA. It is also done by the ribozyme. Also, the removal of intron from the messenger RNA, this is also done by a specific ribozyme enzyme that is called as spliceosome. As well as in the replication of virus RNA, this process is also proceeded by an ribozyme. And protein synthesis also, this ribozyme have rules. Now you have to see that the catalysis of biochemical reaction. As we know that ribozyme are the RNA molecule that possess the ability to catalyze specific biochemical reaction. Also, they play a role in various biological processes that have the transfer RNA maturation, intron splicings, replication of RNA virus, and protein synthesis. So there are some various biological processes in which the ribozyme play an important role. As you can see here, the maturation of the transfer RNA, intron splicing, because the messenger RNA is some non-coding sequence that is called as intron, and this intron is removed by a ribozyme that is called as spliceosome. So the next function of the ribozyme, that is reverse transcription. As we know that transcription is actually the formation of RNA from the DNA. This process is called as transcription. But we have the ribozyme that have the function of reverse transcription. So this is normally, it is about the transcription, formation of RNA from the DNA. This process is called as transcription. Now what the reverse transcription is? So you can see here, and most of the virus, the genetic material, this is RNA. So this genetic material, it is used to convert to form DNA. So in this virus, a process occur that is called as reverse transcription, because in this case is the RNA, DNA is made from the RNA. So this process is called as reverse transcription. Formation of DNA from the RNA, this is called as reverse transcription. Now we have enzyme that is RNA polymerase. It is actually a ribozyme. It's capable of function as a reverse transcription to form DNA. So to form the DNA from the RNA, this process is called as reverse transcription, and this function is actually performed by the ribozyme. So this is the two function of the ribozyme. So we will discuss the application of ribozyme. So the first, you can see here, the ribozyme used as a drug against the virus infection. As we know that virus causes infection in the living body. Now you can see here, this is the genetic material of the virus. This is actually the RNA, though this is the genetic material, and this have the information to cause infection in the living cell. Now we use artificially ribozyme. Now this ribozyme, you can see here, this is the ribozyme. It will make pairing with the genetic material or the RNA of the virus. Now after pairing up with the messenger RNA, now this ribozyme, it is able to cleave this RNA and form pieces. Now you can see here, this is the pieces of the RNA, because this RNA, it is cleaved by the ribozyme. Now after the cleaving of this RNA of the virus, it is not able to cause infection. So this is the practical application of the ribozyme to use as a drug against the virus infection. So this is all about the ribozyme. Thank you so much.